Biology with Dr. E.
The next organelle is the endoplasmic reticulum, or the ER. The ER consists of a series of parallel interconnected flattened membrane tubes called cisterns that enclose fluid-filled interiors. From this diagram, you would notice that the ER is continuous with the outer membrane of the nucleus. The ER exists in two forms: the rough ER and the smooth ER. The rough ER is arranged in folds and its external surface is covered with ribosomes. So if you see in this structure here, you see little dots on the ER. This is a rough ER with ribosomes. The smooth ER is continuous with the rough ER, but importantly, the smooth ER lacks ribosomes. The rough and smooth ERs perform different functions, which we'll review on the next slides. As already mentioned, the rough ER is covered with ribosomes. The rough ER is a site of synthesis of proteins that will be secreted from the cell. It is also the site of synthesis of many plasma membrane proteins and phospholipids. The smooth ER lacks ribosomes on its external surface and plays a role in multiple cellular processes, including lipid metabolism, synthesis of steroid-based hormones, detoxification of certain chemicals, converting glycogen to glucose, and storage and release of calcium. Next is the Golgi apparatus. The Golgi apparatus consists of stacked and flattened membrane sacs or cisterns. The Golgi modifies, concentrates, and packages proteins and lipids made at the rough endoplasmic reticulum. So, how are newly synthesized proteins processed and distributed? This figure illustrates 3 steps involved in protein processing and distribution. In step 1, vesicles move from the ER to the Golgi. Vesicles containing proteins bought off of the rough ER migrate and fuse with membrane on the cis face of the Golgi. In step 2, proteins are modified. Inside the Golgi, some sugar groups are trimmed while others are added. In some cases, phosphate groups are added as well. In step 3, proteins are distributed. Proteins are tagged or addressed, sorted and packaged in 3 different types of vesicles, which bud off the transface of the Golgi. These vesicles have 3 possible fates depending on the proteins they carry. The proteins are broken down into 3A, 3B, and 3C, which we'll discuss. 3A describes the fate of proteins for secretion. Proteins destined for export are packaged into secretory vesicles that migrate to the plasma membrane and release their content by exocytosis. 3B describes the fate of proteins destined for membranes. Vesicles containing lipids and transmembrane proteins destined for the plasma membrane or other cellular membranes fuse with and are incorporated into those membranes. 3C describes the fate of digestive proteins. Inactive digestive enzymes are packaged into vesicles that eventually become lysosomes or fuse with the lysosomes. The next organelle is lysosome. Lysosomes are spherical membranous organelles containing digestive enzymes, also called acid hydrolases. Lysosomes digest ingested bacteria, viruses, and toxins. They also degrade non-functional organelles. Lysosomes also have a metabolic function. For example, they break down and release glycogen. They also break down and release calcium from bones. In injured cells, intracellular release of lysosomal content causes digestion of the cell itself, a process called autolysis. Next, we discuss peroxisomes. Peroxisomes are membranous sacs containing powerful detoxifying substances that neutralize toxins. They play an essential role in neutralizing free radicals. Free radicals are toxic and highly reactive molecules that are natural byproducts of cellular metabolism. Free radicals can cause severe damage to cells if not detoxified. There are two main detoxifiers in peroxisomes: oxidase and catalase. Oxidase uses oxygen to convert toxins to hydrogen peroxide, which is also harmful. Catalase converts hydrogen peroxide to water, which is harmless. Peroxisomes also play a major role in breakdown and synthesis of fatty acids. Let's talk briefly about the endomembrane system. The endomembrane system consists of membranous organelles discussed so far, including the endoplasmic reticulum, the Golgi apparatus, the secretory vesicles, and lysosomes, as well as the nuclear and plasma membranes. These membranes and organelles work together to perform two major functions. One is to produce, degrade, store, and export biological molecules, and the second function is to degrade potentially harmful substances. In this figure, the nuclear envelope surrounding the nucleus is connected to the rough endoplasmic reticulum. Notice the arrow leads from the nuclear envelope to organelles not directly or structurally connected to it, including the smooth ER, Golgi apparatus, transport vesicles, lysosomes, and secretory vesicles.